Hello, I'm audible.
Yes,
Good
yes.
evening, Dr. Harshit.
So we should wait for some time.
Just for 5 minutes only. Let's start.
Sure.
I request all the participants please mute themselves. Okay. Hello. I think we should start. Hello.
Hello.
Yes, yes. Dr. Harshit, I think we should start at the participant join.
Okay. We will start. So, good evening, everyone. I hope you enjoyed yesterday's session. And just a minute. So, I hope you all have enjoyed yesterday's session, and we had discussed the ability of the cells which are needed for them to convert them in tumor or the process of tumorigenesis. Now, as we know, the research is as per the advancement of the technology, research is going on in a very fast pace nowadays, and every month we have so much information, so much data that would take a lot to cover. So instead of covering everything, I had thought that I should concentrate only on one topic, and that's chimeric antigen receptor of T cells, and I should talk about that today in the advancement of the cancer technology. Why I chose this topic is because the first CAR cell therapy has recently been performed first time in India by ECTRIC on a leukemia patient, and that is sponsored by Department of Biotechnology. So we have the thing coming up, and there are some companies which has been established specifically to look after this CAR therapies. There is one company called Immunil, which is specifically concentrating only on this aspect of the cancer therapy. And another reason is this year's last year's Nobel Prize has been awarded for this discovery of this technology. So I'll just start with my slide. So chimeric antigen receptor and T cell therapy. So before I start with so before I start with, I will just go through the basic immune response or basic immunity of human body, so that you will understand better of how this response is being and how this therapy actually works. We have multitude of immune organs and various kind of immune cells in our body, and they together comprise the entire immune system and broadly divided into two category, that is innate immunity and adaptive immunity. So I will not go into the technicality on this. I will just keep the things which refresh your memory and give you a little perspective. You all might have heard the story of Angulimal. So Angulimal was actually a dacoit in the time of Gautam Buddha. So what he used to do, he used to loot the people in the forest when they travel across that forest, and he killed them. And after killing, he chop off their fingers and make a garland and a necklace kind of thing, and he wears those fingers in his neck. So it is a kind of show of that. See, I have killed these many things. So the similar principle is being also applied by our immune cells. So whenever there will be any immune infiltration, we say that if any infection is there or any injury is there, we see the inflammation is going on, and the first responder reaches to that place. So what these first responders do there, those first responders are mostly the phagocytes. They reach at the site of the infection or the injury, and they chew or they eat up the infectious cells. They could be bacteria, they could be protozoa, they could be the dead cells of our own. And what they did after that, After digesting those cells or those foreign particles or foreign cells, they display their protein and their surface. So they start displaying the similar way Angulimal displayed the fingers. So they also displayed that, okay, see what I have eaten today. I have eaten this, I have eaten this, I have eaten this. So when these cells which does this kind of thing, phagocyte, as well as they are called antigen presenting cells because they are presenting the antigen. So now they need the cells and the lymph node. When they reach to the lymph node, various lymph nodes we had in various places, they reach to the new lymph nodes, and there in the lymph node we have T cells present. Now this T cell actually inspect those antigen presenting cells for any foreign antigen. If they have eaten their own cells, those are injured cells, broken cells, so there is no new antigen, all is known thing. And when they encounter any foreign, so now T cell, T1 helper cell, then activate the CTL cell, a cytotoxic cell, natural killer cells, and they reach to the site of infection. And if they found any other infectious organism there, they will kill them by cytolytic process. They also recruit the B cells, and then they pass this information to B cell. B cell then further differentiate into plasma cells and memory cells. That plasma cells start making the antibodies. So this is our immune system. So this T cells not only recognize these cells, apart from these antigen presenting cell, all our cells, our body cells, they also display the protein which they are making. Whatever the protein is manufactured in a cell, the part of that protein is always displayed on the cell surface for the recognition by these T cells. So T cells, when rolling around in the body system, it keep on recognizing whether we have any foreign or unknown protein decoy or protein moiety is on the cell surface. So when a tumorigenesis happen, so some antigen, those are called new antigens because some new proteins are started synthesizing, some new uh, protein expression is being increased, or the mutation is takes place in lots of places. So new form of those protein is being made. So these protein could be detected by the T cells. And when T cell detect those protein, it elicit immune response and it can kill the tumor cell. So if that one is that easy, then how come we have tumor still? If T cell can identify and if T cell can kill the tumor, it can elicit the anti-tumor response. How it is possible that we still have the cancers in human body? So the reason is yesterday when we studied the capabilities or the qualities of the cancer cells, they started working on evading immune system. So they have the mechanism where they inhibit the immune checkpoint, which is another topic completely, where if we go that side is which fall in the purview of immunotherapy. It is also the similar kind of therapy, but it's a difference. So currently that is also a hot topic, that how we can again activate that immune system. But cancerous cells have there is a protein called PD-1 and CTAL4. They have the combination, and until that combination is there, the immune response is not elicited. So when we go, we see this tumor cells are evading. So what scientists has thought that how a cancer can evade T cell scrutiny, specifically if not for the solid tumor, but for the leukemia. because leukemia is a blood cancer, and mainly the cells of immune system are affected. Mm, lymphocytes are getting affected in that, and they all pass through the lymph nodes. Then how come T cell cannot identify them? So the reason is that they do not display that antigen, or whatever the antigen they are displaying, that cannot be identified the T cell. So the next thing is, can we do something about it? So is it possible if we can give T cell, if we tell T cell, it is like that if somebody is going in front of you, and you are sitting and the policeman is sitting, you know that person is a terrorist. Policeman doesn't know he is a terrorist. So you will obviously go and tell the policeman, no, this person is terrorist, you should caught it. So can we do something like that, that we go and tell the T cell that, see, this is a cancerous cell, you should caught it and kill it. And if you kill it, the body will heal. And whatever stress you are facing, it will not be there. So this idea is called uh, card therapy. So this is actually the pictorial representation of how the cells in a cancer environment goes and seen there. So if you see the cancer, yesterday I told you that it is not a homo system. It has a heterocyclic heterocellular system, not homocellular system. There are multiple types of cells there. So we have a macrophage, monocytes, CD4, CD8 T cells, NK cell, DC cells. So these all chemokines which can drive the cells there, and then you have multiple antigen cross presentation by dendritic cells, then T cell and B cell activation by. So this is the anti-tumor environment. This is a tumor where the immune response is there, but sometimes, most of the time, what happens? This inflammation is actually promote proliferation, because our T cell cannot identify it. Only identify the necrosis in between, or inflammation is there. So it start producing more inflammatory compound, recruiting the cytokine and the chemokine, and that leads to the further fast proliferation. So what we do in CAR therapy? So in CAR therapy, antigen receptor is a chimeric. Chimeric means it's a mixture of two. So we modify the receptor which is present on a T cell. So that is why it is called the chimeric antigen receptor which can identify a particular antigen. We are modifying it, and by modifying it, we are enabling it to recognize a particular specific antigen. So it will give, so this therapy fall under a immunotherapy umbrella, and a specific treatment, because it will not, those antigen are not present in a normal cells, only present in tumor cells. So you can identify those tumors, means you are giving them identification. See, this you need to recognize. So there are generations of this, the first generation, second generation, third generation of CAR cell. The first generation has uh, two, the extracellular moiety, the transmembrane moiety, and intracellular moiety, and that is a signal moiety. So which gives the signal for the T cell activation. The extracellular is a receptor variable region, so which can identify a particular antigen, and this transmembrane region is what hold these two signal and receptor together. But when this therapy is being employed, not that favorable results are not being found. So what they did, they actually bought another moiety, and that is a signaling domain. So this signaling domain then gives more modulation and activation to the T cell, and then the success rate is start to increase. And in the third generation, we have the one extracellular signaling, intracellular signaling moiety. So from where we got this receptor? We got these receptors from the monoclonal antibody. We developed a monoclonal antibody against a particular antigen, and from there we will take that specific target. Then that target is then loaded on T cell receptor, and this entire process is done using a retroviral vector. The viral vector is used to transform this T cell, and those T cells then will be injected into the cancer patient, and cancer patient will then respond accordingly. So it has these are the part of chimeric antigen receptor. You have an antibody derived recognition moiety. We have a heavy and light chain of the variable region. Then there is a hinge region, as I said, the transmembrane region. And then you have a T cell activation domain and co-stimulatory domain. And this domain, this structure which you are seeing is an anti-CD19 CAR cell. So this domain is then packed into retroviral gene, and then retroviral gene is then inserted into T cell, and then the T cell then express the particular gene which we want the antibody to be recognized by. In this case, that is the CD19. CD19 is very much, very highly expressed by the B cell lymphoma condition. So this is the first time it has been designed for that. So how it is made? First, they take the first stage is called apheresis. In apheresis, the patient's blood, peripheral blood is collected, because T cell from one patient you cannot give to another patient. So most of the time it is the allo um, uh, pathy. So from the autologous, so from the same patient, you take the T cell, then you engineered it and put it back to the same patient. So there is no cross activation or immune rejection or the self rejection kind of thing we can face. Then we activate the T cell, which we transformed into cytotoxic T cell. We convert them into cytotoxic T cell, CD8 positive cells. Then we transfect those cells with the gene for the specific antigen, and with the help of virus, in this case, the retroviral vector. So you can say the HIV is helping you to kill the cancerous cell. Then we expand the cell. Those cells which are transfected, those will be cultured, expand, and proliferate, so that we have an ample amount of cells to inject into the patient. Just so then conversion cells will be purified and cryopreserved, and cryopreserved for the further, and they can also be freshly infused into the patient, and they infuse in the patient, and they will show their effect. So this is the pictorial diagram of the same thing, where the PBMC, peripheral blood monocytes, are collected from the for genetic modification. Then those will be activated for the, and they converted, and then anti-CD19 CAR therapies be given. And before that, patient is also given lymphocyte depleting chemotherapy, so that it will not have any lymphocyte which will affect this process. So this is how this is pictorial diagram how CD19 particularly works. So in this, you have a transmembrane and this linker. So this is held by the uh, hinge joint linker, and then the co-stimulatory and stimulatory domain is from CD3 gamma, which is taken from the B cell, and it is specifically to activate the cell. So the co-stimulatory signaling then stimulate the T cells, and it also got phosphorylate with the help of Z70 protein, and the signaling cascade then further activate the transcription of T cell effector function. So those T cell effector function is IL-2 secretion, proliferation, cytotoxicity, and then they create actually the perforin pores. They create pores into the tumor cell after recognition, and the cell will lead to the cell death. The types of CAR cell therapy, the different CAR cell target different specific surface proteins. So the surface protein must will be selected on the basis of how much it is expressed by the cancer. It should be highly expressed. If it is a new antigen, it is much better, and it should be have the very high expression level, and it should not be expressed by somewhere else in the body, so that there will be minimal side effect to be seen. And so most of the targeted cell receptors which are used antigens, those are CD30 in the case of Hodgkin's lymphoma, BCMA for multiple myeloma, SLN F7 for again for multiple myeloma, and CD19 is used for the lymphoma and ALL cases. So next is another thing. Is there any therapy approved for the treatment? Yes, there has been approved. The first such therapy is been approved in 2017 by FDA, and so that's first FDA approved CAR therapy targeting CD19. Its generic name is Tisagenlecleucel. It's very difficult to pronounce. Chimeria, it's a name. It is approved for the in children and young adults with ALL. The another therapy which is recently approved again in 2017, anti-CD19, same for the replaced BCL, Excaptazine Silolucel, or Yescarta is a name. And then in 2018, they again approved Chimeria, the same thing for the non-Hodgkin's lymphoma. So there we can see the furthermore CAR to become. So what we had in India recently acted, that is CD19 and CAR therapy. So there has been lots of side effects. Some are very common, some are very rare, but these could be seen because it's the immune response. When you increasing the immune response, all immune related symptoms or immune response symptoms can be seen. So cytokine release syndrome, which is characterized by symptoms similar to sepsis due to infection or severe flu-like syndrome, like high fever, tachycardia, hypoxia, hypotension, decrease in a liver kidney function, prolonged PTT, and risk of bleeding. And sometimes the patient is also required ICU admission. So it is similar as what is seen in a very severe cases of corona also, because the corona cases also, the first 5 days it's the viral days, after 5 days it's immune days. So when immunity goes berserk, then the similar symptoms are seen. Apart from them, there is a neurological toxicities are also be seen, such as confusion, somnolence, tremors, gait instability, aphasia, and other difficulties in speaking, seizures, and neurologic toxicity. However, these toxicities are not that life-threatening or give that much problem. They can be symptomatically addressed by the clinician given case-to-case scenario. And so CART is comparatively much more safe and better uh, than the generalized chemotherapy, which is being administered routinely. So today, I think I finished quite early. Yesterday, I was worried. Sorry. So actually, I wanted to keep the little time. If there has been any question or discussion, we could have. I don't claim to be the expert and know everything, but I might learn something from all participants also.
Uh, Thank you, Dr. Harsh. It's a very nice and informative presentation. I hope the participants liked it and they gain something from this presentation. Uh, I actually asked the participants if they have any questions, they can ask to the Dr. Harsh. They can unmute themselves.
You can ask whatever the question you have in mind, or if you can't speak.
Hello, sir. Am I audible?
Yes, yes, you are audible.
Okay, sir. Actually, I asked you yesterday regarding the cardiac cancer
Yes,
and regarding the immunotherapy in cancer. So, what will be the role of the residential T cell? Because nowadays it is being very prevalent regarding the residential T lymphocyte.
yes, residential T lymphocyte. Actually, it is being seen. There are two scenarios is given. One is where the tumor is developed in an immunocompromised. In a, I'll give you example from the mice. When tumor is developed from the immunocompromised mice, and its immune cells were taken and used, they cannot destroy the cancer. While the if the tumor is grown in a immunocompetent mice and the immune cells taken from that mice and used, they be able to destroy the cancer cells. So resident T cells, yes, they have, as I said, that heterocellular environment is there and T cells are got primed and they express. So their number is minimal. So nowadays people are trying to explore how more they can get the use of those because certainly they are having the expression they got interact with the antigen presenting cells and they are also present in the vicinity. So it's a role is again the same that provide the immunity, but since their number is very less, so they cannot give that much level of the immunity defense from the to the tumor.
Okay, thank you, sir. I have another question. Should I ask it?
Yeah, yeah, sure, sure.
Okay, okay, thank you. Regarding the CAR-T immunotherapy related to cancer, so what will be its propensity regarding the treatment of cancer stem cell?
Cancer stem cell is a complete different kind of you know area. We still have a debate in a scientific society that whether to call them a stem cell or not. Certainly, they express the stem cell marker and they also show the plasticity, they show the colony formation. So CAR therapy actually does not distinguish between whether it is a stem cell, whether it is a, because it's a T cells. You have given it a target that this is your antigen, and if you are finding that antigen, you go and attack it. So it is a kind, we have seen the autoimmunity where the T cells can chew up the entire bone marrow, T cell can chew up entire an organ. So they don't differentiate between the stem cells and non-stem cells. So CAR cell therapy will work against if that um, particular antigen is being expressed on those cells. Thank you, sir.
Hello.
Hello.
I'm audible. Na?
Yes, yes.
One more question in the chat box. For CAR T cell therapy, are the cytotoxic T cells used only or any T cell? This is question from Anindita. I am I think there's a network issue. Hello. Am I
We have
audible? Hello, Dr. Harshit, can you hear us? I think there is some network issue.
sorry, actually there is electricity problem, so my
Yes,
internet got disconnected. Can you repeat what you were saying? I just connected it to my
CAR
phone.
for CAR cell therapy, are the cytotoxic T cells used or any T cell can? The question from Anindita.
No, no, they actually, when they take the peripheral blood, they collect the entire T cell milieu and then they activate it, converted them, even if whether it is a T helper cell, they convert it to the cytotoxic cell because their job is to kill the cell. So they only use the cytotoxic cell, not T helper cell or not TH1 or TH2, not T immune cell.
Well,
Hello.
yes, you are audible.
Yeah. Yeah. So any other question? And you can write in the chat also if somebody want for CAR cell. We are the cytotoxic cell use. Again, this is what the question you had asked. Any other question? Anybody?
Yes, it's over. Okay, thank you so much, Dr. Harshit. It's a nice and a very informative session. And I, from Sage University, I thank you so much from the precious time you have just given us such an informative session. And not the least, but the participants who are here present and patiently listen all the sessions. I thanks all the participants to be present here. And if they want to share anything they can be, they are welcome. We can share any. We have only 2 minutes left with us. So if any participants want to share anything with us, they can be welcome. So if anyone.
If you have any feedback for me for my improvement on presentation skills or anything, you are all very much welcome. And or if you have any question or any other thing too, you just pass on to Varsha. She will communicate with you. Thank you very much, Varsha, for inviting and giving me opportunity to speak. Thank you very much.
Thanks. Thank you for accepting.
Of course.